Thank you for the introduction, and hello everyone. My name is Xueyi Dong, and I'm a postdoc at Weihai in Melbourne, in Australia. And I'll talk about benchmarking long-read RNA sequencing analysis tools using in silico mixtures. This is a published work, so feel free to take photos. All right, so let's begin. When people are saying RNA sequencing, usually by default they are referring to the short-read RNA sequencing, which they use the second-generation sequencing technology, and it's also called next-generation sequencing. And using this sequencing technology, the reads are the RNA or molecule will be fragmented during the library preparation. So actually the data they get are the fragmented pieces of the transcripts, which means due to the read length limitation of the technology, which means you are not able to get the whole read that is covering the full transcript. And for analyzing the short-read RNA-seq data to quantify the amount of expression level of each transcript or each gene, usually the first step of the analysis is to map the reads to a reference that can be like a genome or a transcriptome. So by this way, we can know where does the read come from the genome or the transcriptome. And then we can perform counting, which is counting the number of reads that are overlapping each region of the genome or transcriptome so that we can estimate the expression level of genes or transcripts for all the genes or transcripts in each sample, and so that we create a count matrix. And because the reads from short-read sequencing technology are having a limited read length, while some genes may have alternative splicing happening, which means one gene can produce multiple transcripts, and the transcripts may have some parts of the transcripts overlapped. But for short reads, usually the reads are just overlapping the exons, and they may be mapped to multiple transcripts because of their overlapping parts. So for the transcriptome mapping using short-read technology, there may be some mapping uncertainty due to that. So there may increase the technology noise of this kind of quantification methods. But now we have long-read RNA sequencing technology, and it's the third generation sequencing. Well, nanopore sequencing is one of the long-read sequencing technology. It works by having the RNA or DNA molecule going through a biological nanopore on membrane and records the electricity signal to detect each basis. So theoretically, there is no limitation for the read length. So we can possibly get read that is covering the whole transcripts, and we can get in the reads we can also obtain the exon junction and also the exon chain information. So it can help us getting better quantification for transcripts. However, comparing to the short read where the sequencing accuracy is pretty high, the long read sequencing technology has a slightly lower sequencing accuracy, which means there are more sequencing errors in the reads we get from long read sequencing technology. And also the number of reads that each sequencing flow cell can produce are also lower in the long read sequencing technology. So which means if you don't use a high number of flow cells to produce the data, then the number of reads you can get from each sample will be lower, and which may impact the power of your differential analysis. So this means that some pre-built tools for analyzing the short-read RNA sequencing data may not be suitable to use on long-read sequencing. And in this analysis, our aim of the analysis is to benchmark the tools that can be applied on long-read RNA sequencing data analysis. And because there are quite a lot of steps in the typical transcriptomic analysis workflow for long-read RNA-seq data. But here in this study, we were just focusing on comparing the methods for isoform detection, which is detecting whether there are any novel transcripts that are not annotated from the in the sample. And also we're gonna compare the methods for the downstream analysis, like differential expression and differential transcript usage analysis for the transcripts, which are more like focusing on the splicing, alternative splicing. Cool. And then, yeah, so to compare the methods, we need to use a specific dataset design to ensure that there will be ground truth so that we can know what is correct, what is wrong, and what is good, what is bad. So we need to include ground truth in the dataset design. So to include ground truth in our dataset, the first way we did is to using sequence, which are some synthetic spike in RNAs that are like artificial RNAs from artificial genes they designed. And some of these genes are designed to have alternative splicing, and the transcripts are already mixed into known proportion into two mixtures, A and B. And because all the isoforms in the sequence are unknown, and the abundance of all transcripts are known, and we know which ones are the real differentially expressed ones between the mixture A and B, so we can use it as one way to introduce ground truth to our dataset design. So in our dataset, so we choose two human lung adenocarcinoma cell line, and we added the sequence mixture A and B into each of the cell line. And for each of the group, we have three technical replicates. And for the sequencing, we use both the Illumina short-read sequencing and the nanopore long-read sequencing to sequence the materials. And after getting the pure cell line and the sequence samples, we further did in silico mixture to create the in silico mixture samples by downsampling the reads from the pure sample and then mix them together. So in this way, we also introduce ground truth from the cell line, even if from human cell line, we don't know which genes that which transcripts are actually differentially expressed between them. So that's how we use it to introduce ground truth. And after that, I'll talk about the results. And firstly, here's the analysis workflow. So for isoform detection tools, we just use the pure RNA samples, and we choose 6 different tools to compare. While for testing the downstream analysis tools, we mapped and quantified our reads using the reference transcriptome and using the some well-performed tools that we chose. And we using this counts, and we compared 5 different differential transcript expression analysis tools and 5 differential transcript usage analysis tools. And here are the analysis results. So first of all, here's the data quality summary. Because we used 5 flow cells for our nanopore sequencing, so we get a pretty good sequencing depth. So we get around 40 million reads per sample, which is much higher than most of current nanopores RNA-seq datasets. And for next, we go to the results for the isoform detection tools. And here we use the sequence because the sequence are the artificial transcripts. So everything from the sequence genome should just coming from the annotated sequence. So we can use it to know which are the true discoveries and which are the false discoveries. So we can calculate the precision, recall, and from here we found out that the tool called Bamboo outperformed the other tools because it's having higher precision and recall. And also we did some other analysis on the isoform detection tools, and we found out that not only Bamboo, but also StringTide 2 are also performing quite well. So we believe these 2 tools are outperforming the other isoform detection tools. Okay, and then let's go to the results for the comparison of differential transcript expression analysis tools. So here I'm showing the number of detected differentially expressed transcripts in each comparison. We can see that in the comparison of the between the 100 versus 0, which are the pure samples, we get the highest number of differentially expressed transcripts. But for the comparison between the mixture samples, the number of DE transcripts we detected are decreased because the fold change in them are getting smaller. And in this plot, we found out that Lima, HR, and DSIC2, which are the three gold standard methods, seems to perform quite similarly. And in the left plot, it seems like the tool called Noisic has a pretty good sensitivity. But when we look into the sequence and we calculated the false discovery rate, we found out that Noisic has a bit high false discovery rate. And we also calculated true positive rate using the sequence back-in, and we found out that here Lima, HR, and DSIC2 also performed quite similarly, and the true positive rate of them are quite well. So overall, we think DSIC2, HR, and Lima are the outperforming tools for differential transcript expression analysis, where HR slightly outperforming the other tools in the control of false discovery rate. And then let's go to the results for the comparison of differential transcript usage analysis. And here we use the sequence to calculate the false discovery rate, and we found out that from all 5 tools we compared, only HR has controlled the false discovery rate well. But when we look at the true positive rate, we found out that for HR, the true positive rate for the comparison between the mixture samples decreased quite a lot. So it is not that sensitive. So this suggests that for all of the tools we detected, there is no one that balanced the accuracy and sensitivity, which suggests that there is more room for developing better methods. So this slide shows a summary of the benchmarking results. So briefly, in summary, we designed a special dataset for comparing long-read RNA sequencing data analysis tools, and here are the results we get. And finally, I would like to thank everyone who contributed to this project. And if you are interested, you can look into more details by scan the QR code and look at our paper. And you can also see me at the poster session. So I have another poster today later on. And I would also like to thank ISCB and the Weihai Postdoc Association for providing the traveling scholarship to support my travel to this conference. I also like to thank the conference organizing committee. Thanks, everyone.
Okay. Thank you for a great presentation. Do you have any question from our audience? We don't have online question. If you have a question, you can come over.
Well, thank you for the talk. It's good to know which one is performing better than the others. But do you have any idea why they are performing better? Yeah, thank you.
Sorry, just which part of the analysis are you talking about? For Bamboo, yeah, because Bamboo is a reference-based tool, so it uses the information from the reference. For example, which transcripts are there inside of the annotation, and also like they have the junction information that are already annotated. So that's maybe part of the reason why it's helping to like helping Bamboo to get a better result. And also we found out that for Bamboo, they output all the transcripts that are provided in the annotation into their discovery. So maybe that's part of the reason why we're getting a higher higher recovery rate from Bamboo.